Hello, this is BrainCan channel, the channel about brain research. Thank you for watching us. Don't forget to subscribe and don't forget to like this video. So, if you will ask me to describe the brain activity, the neuronal activity in one sentence, I would tell you something that it is strictly organized chaos. Also, it's quite similar to music. So in music, you have sounds and you have pauses. And if we come back to neurons, neurons also have periods of their firing, of their very active neuronal activity, and they also have periods of silence. And this silence is very critical to encode information. And there are different mechanisms how to modulate this silence in neurons, and one of them is based on other neurons. So in the brain, the majority of neurons are excitatory, but there are also a proportion of inhibitory neurons that actually suppress activity of other neurons. But what if you are not a neuron? Is it possible to modulate the neuronal activity? Not so long ago, it was found that it's possible to modulate neuronal activity with perineuronal nets. And today we have a special guest from Charité Hospital, and correct me, okay? From Berlin, from Institute of Neurophysiology, Luna Šošo Zdravković. She is doing her PhD and she studies perineuronal nets, microglia, and much more. And today we'll talk with her what perineuronal nets are. So Luna, could you please tell about history of these nets? What is it, and so on?
Sure. So it's one of the aspects of my research that I focus on. So these perineuronal nets, as the name suggests, they're a type of net-like structure, and they basically they exist around certain neurons in the brain. They were actually known for a long time as just a structure that was described even at the end of the 19th century. So the well-known Italian biologist Camillo Golgi has seen them first time and described as a sort of a net-like, spider web-like structure around the cell bodies of certain neurons. But what is really interesting, I think, is that the function of these structures was only started to get elucidated end of 90s, so 20th century, basically only 3 decades ago. So what these structures are basically, they're a component of what we call the extracellular matrix. This is just the environment of these proteins and various molecules that float around neurons in the brain and surround them and give them support and structure. And so these perineuronal nets, basically they enwrap around the bodies, which we call somata, of neurons, and around some of their proximal dendrites, which are basically the input regions where the neuron receives information, as well as part of the axon, which is their output, where they send off information further.
So it's not just a mechanical support of the cell.
Exactly. So although it's still like a bit of a black box of a structure, so we have several ideas, and in the research field there are several ideas of what these structures really do. So the most fundamental idea is, sure, they do provide a structural support and a bit of a shield. So it's always good to look at where these structures are, and is there a specificity of a type of neuron that they surround, and funnily enough, there is. So they tend to surround these special type of neurons. They're inhibitory neurons, which you already mentioned, and they are what we call parvalbumin positive fast spiking interneurons. So
Very active, guys.
Very active. So these are so let's break down that name. So the parvalbumin positive. This means this is like a special protein that is only characteristic to these neurons. You cannot find it anywhere else. It's a for those of you who are more interested, it's a calcium buffer, and calcium is very important for activity.
For communication between the cells. Yeah.
Exactly. So they're parvalbumin positive. That's their marker. That's the marker protein. That's how we find them. When we want to stain them, we use parvalbumin. And then they're fast spiking, which means they have this ability to produce action potentials. So to change their memory potential from their resting and to send a signal off in the form of the action potential. So they have the ability to do this 300 to 600 times a second. So it's a very high frequency of activity. So that's why we call them the fast spikers. And apart from that, they are functionally very significant. They act as a master controller of broader network activity in the brain. Even though they're in numbers, funnily enough, much less common than excitatory neurons. So as I said, it's very important to look at where these perineuronal nets are, and they happen to be around the bodies of these neurons. And so the structural support they give, at least this is the hypothesis, is since these neurons are so active and therefore require so much energy and sugar and oxygen, exactly, to create all these spikes. So the nets are there to keep their tiny little environment around the neurons constant, because these neurons have to have a constant inflow of oxygen, of sugar, and to support this activity. So they are very sensitive. So they need this little like a shield around them. So that's the function one of perineuronal nets, like a structural support. The functional aspect of perineuronal nets is very interesting as well, and this is the part that got sort of elucidated end of 90s, early 2000s.
Do you mean neuronal plasticity?
Exactly. Okay. So they're basically, we call the perineuronal nets the guardians of plasticity. So why? What does this mean? So when you develop, for example, when a child develops, there are these critical periods of time, which we call critical plasticity periods. And these are the times where the brain is very malleable. So it's very malleable to the influence it receives from outside. The neurons, the connections between the neurons are very, we call it plastic. So
they can remodel quickly all the synapses and spines that we can find in neurons.
Correct. And so so the brain is kind of like a sponge. It really takes in all the input, and based on this input, it wires and rewires and slightly adjusts itself. So what is very interesting, you have this timeline, let's say for a human child, up to about 2 years of age, the brain is highly, highly plastic. And it really relies specifically, let's give an example, specifically for visual inputs. It really relies on visual cues. And then from the age of 2 to approximately 8 years old, this plasticity sort of starts to die down a bit gradually. And this coincides with the emergence of perineuronal nets around the neurons that I mentioned already. So the emergence of these nets starts around 1 to 2 years of age.
I have a question about this. Since they provide mechanical support and they shield neurons, is it possible that they restrain actually neurons? So they prevent this quick remodeling of synapses? It
could be. It may well be, because you mentioned at the beginning, we were talking also about the interactions of neurons and non-neuronal stuff in the brain.
Yes.
So there happens
to be about half of other cells in the brain.
It's not even half. It's actually 3 times more than the neurons.
It's dependent on the matters, yes, how you look at this. Yeah.
So there are these other non-neuronal types of cells, like, for example, microglia, like astrocytes, and they all come into contact with neurons. And there is a hypothesis indeed that since these nets are around these somata of the cells, the cell bodies, and the proximal parts of the dendrites, where also the synapses are, that they maybe prevent excessive influence from other types of cells in the brain and also from other neurons forming synapses onto these cells. thus making these cells that have the net around them more stable. And so this is like when I mentioned this visual input paradigm and how the child develops and how the nets develop in the child as it receives visual inputs. From ages 2 to 8, you have a progressive development and complexity increase in the perineuronal nets. And this stops the wild neurons from being plastic and
uncontrollable.
Yes.
So very important point. If you want to work efficiently, then you have to have some periods of non-working,
Exactly, and you need to actually have a focus, basically.
Yes, exactly, yes.
This is what I see perineuronal nets as. They don't necessarily only limit the neuron, but they focus it, because you kind of, as you develop, you go towards specialization, and this brings more efficiency to the entire system. So yes, so this is another function of perineuronal nets, the most important with regards to plasticity. I mean, there's a fine example, like, for example, children who have a lazy eye, for example, what they do is they put a patch over the stronger eye, so that the lazy eye can get more input while the brain is still plastic. And train. And so this can happen only in this critical period between the ages of 2 and 8. After that, for example, you could not easily do this, as the perineuronal nets have set in. They have ensheathed the neurons, and there is basically no change. But when you think about that, then there's potential. There's a thing in the field at the moment of perineuronal nets where people are thinking, can we somehow change or remodel the nets, so that we can reawaken this potential, this plasticity that we have as a young child? So, for example, the potential to learn languages so easily, which is another thing tied to critical periods.
I know it. I know it.
Can we reawaken this in the adults? And then, there's also a hypothesis that it could have a beneficial effect for things like disorders like PTSD, for example.
Exactly.
So it's very interesting, I think, topic.
What is the source of perineuronal nets? And as I remember, but I can confuse, that this is polysaccharides, right?
So this is one of their components, indeed. So the nets, maybe this was an important thing that I missed in the beginning. But what they are composed of is, so they have a core, which is like a scaffold of a protein. Typical proteins are aggrecan, chondroitin. So these are all the proteins that are also found in this extracellular environment of the neuron.
Matrix,
And then these proteins, what you have connected is these sugar-based molecules, but it's basically oligosaccharide residues, we call them. And they attach to various parts of these proteins and form this
polymers. Because they're
exactly.
polysaccharides, they can form nets, these lines, and it looks exactly like a net. So imagine a neuron in a net.
Exactly.
like a fish, for example.
Like when you have the how do you call it? Like a net, in it you have oranges. This is how it looks like. It's like exactly a net, like
this back.
Exactly. So it's mostly like a mixture of these proteins. There's specific groups of proteins, to get maybe more technical. They're called CSPGs, which stands for chondroitin sulfate proteoglycans.
Stop saying this.
Yes. Sorry.
Minus 300 subscribers now on my channel.
But that's what they're composed of, basically. So a bit of proteins, a bit of sugars, and based on this, actually we also try to identify them by finding whatever chemicals bind to either the sugar parts or the protein parts. Then this is how the labeling of perineuronal nets is managed, basically. You either find lectins, which are proteins that bind to sugars, so you have a labeling of this sugar part, or you find proteins, which is antibodies that bind to the core proteins of the nets, and you have, again, another net labeling.
So there are different nets in the brain, but they're mostly formed around paralbumin positive neurons.
Basically,
or
exclusively. It's very rare to find them around any other type of cells. It's in my experience, it's something like 10%, maybe at maximum, of any other cells that have these nets. And for that tiny percentage of cells, it's not even really known why they need them. Because, as we discussed, for the probably positive neurons, function-wise, it makes sense. So,
Exactly.
But for these other types of cells, it's a bit of an unknown for now.
And I think now we will step into a very dark side of this research. All compounds are synthesized somewhere in the body, and what is the source of perineuronal nets and
This is really a good
proteins and these polysaccharides? Can, for example, any cell express the protein that is part of this net? Or it's cell specific somehow?
This is a really good question, especially since the perineuronal nets emerge during development, which is also another under-researched part of the developing of the brain. We know a few things. We have a few breadcrumbs that can kind of lead us to a somewhat of a picture of how this happens. So the neuron itself that wants to, let's say, have a net around it needs to have a few enzymes that synthesize at least the core proteins, it seems. But then there has to be apparently a contribution by oligodendrocytes, which are the cells that form the myelin sheath around neurons, helping them conduct action potentials quicker and more efficiently. So one component would be oligodendrocytes, and another would be astrocytes, which is another non-neuronal cell type in the brain, very versatile, very much involved in synaptic transmission and many processes that support the neuron, nourish the neuron, help it recover from injury. So astrocytes and oligodendrocytes are mostly implicated. It seems like microglia, another
adorable microglia,
another glial cell type, is
we are working on.
Exactly. So full disclosure, I'm a microglia researcher, so they're my favorite. So they also seem to be implicated in generating the perineuronal nets, although rather in this element of remodeling them.
Because perineuronal nets are also a very plastic system. Yes. So they can be formed, but they also can be digested from some side. And microglia is the typical macrophage.
Yeah. Yep.
Can come, digest part, then we have a free synapse, and it works.
Exactly. So microglia are sort of perfect for this, since they are basically the brain's immune cells. So they have this ability to chomp off parts of structures like synapses, and it seems like, as the neuro research shows, they can also influence the remodeling of perineuronal nets. And the perineuronal net itself, as you mentioned, it's very cool. I think that it's a plastic structure. Exactly. It's like when we think of the body's skeleton, we always think of it as a static structure, but even the skeleton constantly gets remodeled. It's constantly degraded, built up by various types of cells that kind of chip it off and then give it more bone back. And so the perineuronal nets are much in the same way. So they're very mobile, let's say, structure in motion. And what is also very interesting is I think that they seem to be very much influenced by the environment that they're in. And for example, since I work with brain slices usually, so with acute tissue, they seem to be influenced by the quality of the tissue. So so this is...
I guess region of the brain.
Exactly. So in different regions... In different regions, they can look very differently, and also in different stages of life. When they start to emerge and when they're still forming and piling up all these chondroitins, all these sugars that make up the perineuronal net, they can look strikingly different than when you have an older, let's say an older sample. So there's that, and there's also the aspect of pathology, which is, of course, very relevant to everybody. In science, it's always the hot topic. So
what happens with aging, for example?
With aging, perineuronal nets they don't change too much. So one trend that can be seen is they just become more dense. It seems like it, so more stable.
But it's difficult yet to make any conclusions, right?
Yes.
Because this is still a very
young field, I would say. Sure, 30 years, but it's still, especially this pathology aspect has only now started to be really explored with this context of perineuronal nets.
And neuronal plasticity.
Exactly. So, because plasticity was, I think, at least this is my theory, why perineuronal nets got so much in the spotlight in the late 90s was because plasticity was all the rage in neuroscience at the time.
Yes.
So it was a hot topic, and this was kind of adjacent. Somebody probably did some stainings to, complement functional data and saw this dynamic of perineural nets.
Do we have some contradictory questions in this field, for example?
Contradictory questions? Well...
let's pick up one.
Yes. Contradictory questions. So there's a few things maybe. Maybe the question of localization in certain places in the brain. So it's not that all PV positive interneurons have the nets.
Wow.
So some of them, a lot of them have it, but not all of them. And it seems to be like, for example, in neocortical layers, so in this newest part of the brain, the kind of the peel of the brain, you can actually see that there is a discrete distribution, in fact, of perineuronal nets only around certain PV interneurons and certain locations, which are layer specific.
And I think the reasons why it is sudden are not known yet.
It's not known. It's absolutely not known. So this is just something that has been very recently described. So you have, the neocortex has 6 layers. Different layers basically are different inputs or output zones that then carry on towards other parts of the brain. And so you have like a different arrangement of cell types, not just neurons, also glial cells. And then you have different types of neurons as well by layer. And it seems like that these probably positive interneurons that are in the deeper cortical layers, they tend to mostly have the nets very well developed. And their axons are also very branched, so there's a lot of innervation going on there. But in the higher layers, like layer 2, 3, they don't seem to have they either don't have a net or they have a very thin, like a veil, like a net. So this is also a gradation that is very interesting to see. Perineuronal nets are not always like this strong net built around like a fortress around the neuron. So they can also be very thin and veil-like. And it seems like there's a correlation between the complexity of the net around the neuron and the neuron's physiology and function. So this is very interesting. This was already studied. There is one very cool paper. I actually really like it. Usually, if you want to study something in science generally, you either have to look at the baseline or you have to perturb the system and completely turn it upside down somehow, either knock something out or take something away.
Remove one factor, yes.
Exactly.
Check what happened.
And there is this it's a bit of a rough approach. So there is a way to completely remove perineuronal nets. There is an enzyme. It's called chondroitinase. It degrades all the nets, really like strips them away fully. And basically what happens then to the neurons themselves, the PV interneurons, is when you check their electrical properties, which is what we do in electrophysiology, you can tell that the properties have completely changed. The kinetics of their action potentials are different, slower, they're more excitable. So these are all the things that it seems like it's kind of a hint that the perineuronal nets might be involved also in these functions and conferring these functions to these PV interneurons. So,
thank you very much, Luna.
Yep.
Good luck with your research.
Thank you. Thank you for having me.